Good evening. I am Tapaswini Sharma and I will be speaking on behalf of my team, Aegis. In Greek mythology, Aegis was the shield wielded by Zeus and Athena. It was made of a divine animal to provide protection. We have come up with a solution to protect astronauts from radiation in space and make space colonization safer. Next slide, please. I will be talking about the problem, the solution, the risk analysis, the experiments, and the conclusion. Next slide, please. So talking about radiation. Radiation is basically energy packed in electromagnetic waves or carried by particles. It can penetrate habitats, spacecraft, equipment, spacesuits, and can seriously harm astronauts. It is a serious problem that can cause damage to all parts of the body, including the central nervous system, the skin, the skeletal system, and the blood-forming organs. It passes right through the skin and sheds energy there. It also fragments cells or the DNA. The surface of the moon is badly exposed to cosmic rays and solar flares, and some of that radiation is very hard to stop with shielding. Furthermore, when cosmic rays hit the ground, they produce a dangerous spray of secondary particles right at your feet. All this radiation is boosting the risk of cancer and other maladies. NASA has proposed some solutions like using aluminum or lunar water. But radiation shields would need to be very thick in order to provide some protection. Thus, they would result in heavy weights and high costs. Next slide, please. So the solution. Now, we aim to solve this problem with the help of extremophiles. Aegis is a physical shield with inserts of radiation-resistant material to protect the habitat. The radiation-resistant material will be made from the extremophiles. They are microorganisms which can withstand the radiation in space. So extremophiles have enzymes that protect them in space, called extremozymes. We will be extracting the enzymes that absorb the radiation. Now, after extracting the enzymes, we will use them to create what we call the ink. This ink will then be printed onto the material. This material will be a 3D printed shell or wall. It can be silicon derived from the lunar regolith. So we can form a structure with lunar regolith in some manner, such as 3D printing. Next slide, please. The risk analysis. There are a number of risks associated with a dome shield for radiation protection. I will be addressing the two major areas of risk here. The first is structural risk. During the construction of the dome, we don't want the dome collapsing or any equipment failure during the construction. These risks can be mitigated by careful design and testing of the shield construction process prior to construction around the habitat. Initial testing will be done on Earth and using computer simulations. Final testings can be done later on the lunar surface, constructing a scale model of the shell. The second area of the risk is planetary protection and contamination. So there are international guidelines for planetary protection, which I think every country has their own interpretation of. We will have to go with the interpretation of the one who will be building this in terms of the documents required, what sort of paperwork is needed, what registrations have to be done, and such to get their approval. Because we need approval to carry the various biological elements. So the biological agents used to create the radiation-resistant materials are proposed to be extremophiles. Extremophiles can live in the vacuum and radiation environment on the moon. Therefore, they will need to be contained to avoid contamination in line with the planetary protection guidelines issued by the state or the states conducting or licensing the mission. There will be oversight by the relevant agencies in the licensing state, and the project will need to conform with the safety standards. No proposition on the method of meeting these requirements is proposed herein. The actual mechanisms for doing so will be addressed through rigorous future testing. Next slide, please. Now for the experiments. The experiment is to test the different extremophiles against ultraviolet radiation. There are a number of risks to the people who are conducting these experiments. This involves the UV radiation risk and also the risk of contaminating their environment with extremophiles. Having a shield covering the experiment when it's being performed would mitigate the UV radiation risk. And for the other, we propose using a glove box. You can get a portable ones for about $1,500 to $2,000 that will allow you to do your experiments in a sealed glove box, and be sure to clean inside of that to remove any extremophiles prior to actually opening the glove box to remove the components. In case you have the apparatus, the cost for the microorganisms would be around $600. But the rewards are knowing how much radiation these particular extremophiles can resist. And that is the first step to being able to create our own shield based on the extremophile that would protect the astronauts in their habitat on the moon and possibly beyond. Next slide. In conclusion, this project, Creating a Shield, is a long-term, multi-year project, but it's an opportunity for the Asclepios team to carry out fundamental research that could shape the future of humanity in our solar system. Next slide, please. So before taking any of your questions, I just want to mention that although we were in completely different time zones, we created something that we are happy with. This has only been possible because of my hardworking team members and our amazing mentors. Thank you again.
All right, great job. Okay, at this time, I would like to invite the mentors to ask the initial questions, after which we will take questions from the students. Is there any mentor that would like to ask a question?
Sorry, I have a question. I'm not sure if you said that, but how would you provide with the extremophiles? Can we buy them, or how do you get them?
So the extremophiles are actually available on several websites as well as some other stores. We have provided this in our detailed report, okay. But yes, they are available.
Okay, and is it cheap or expensive?
We have taken the average cost of 3 extremophiles, which will be the most helpful in protecting from radiation. So the average cost is about $600.
Okay, thank you.
Thank you.
And do these extremophiles need special living conditions during transportation or anything like this?
Well, considering they're extremophiles, they can survive anywhere. But we would suggest keeping them in a glove box so as to not contaminate the area that you're working in. If you already have one, well and good. If you don't, you can get it for around $1,500, and those are portable, so you can carry them around.
Okay, thank you.
Thank you.
I have another question, sorry. So we know that the extremophiles absorb the radiations and they can live with it, but are they going to block the radiations and thus enabling the layers, the shield, to be really efficient?
So the best part is that they have enzymes which actually absorb radiation, and there are also some extremophiles that are very good at doing so. So we will be using these enzymes and we will be combining them with some other materials to create the shield.
Okay, thank you.
Thank you.
Hi, thank you for your presentation. It was really interesting. I have another questions too. So considering the next step of your project, if I understand correctly, would be to establish the experiment that you presented in your last slide. Would that be an experiment that will have to happen in a lab, or would you see it during the analog mission itself?
So we have made this to be incorporated in your mission for the analog astronauts. So this can be incorporated, and it's something that we think is going to be very useful. You can get the portable glove box, and that can help you a lot with the experiment. Does that answer your question?
Yes, perfect. Thank you very much.
Thank you.
I also have a question, but it's a little big one, so please bear with it. Being a microbiology guy, I just wanted to know that if you're carrying a living organism into the space, they need a material medium for staying alive. Okay, so if we are not keeping them alive, then the enzymes inside them aren't going to work. So if you have anything uh, that you are going to replace every time once the materials get exploited or depleted.
So our current extremophiles that we are suggesting are tardigrades, and tardigrades need fluid to survive. So they suck juices from algae and such, and we can provide that on the lunar surface. Apart from that, in case they do not survive, I think even changing the what what can I say, the enzymes would be quite cheaper than what we are currently doing.
Yes, as far as my understanding goes, uh, the radiations are harmful to the organisms primarily because they attack the DNA. Okay, so the DNA chain is broken down, and because of that mutations takes place, and that is the primary cause of the radiations over an organism, over every organism. So the extremophiles have a number of their own DNAs, so that one DNA gets destroyed, they have another one, or they have a DNA repairing mechanism, but I don't think they have any enzyme that actually bears the radiations rather than as far as I know. So, is there any enzyme actually that bears the radiation and stops it from going through?
Could you please repeat your question?
Okay, so as far as I know, the radiation primarily harms the DNA structure and not any other things. Most probably, the primary reason is harming the DNA structure and mutating the organism because the DNA structure is broken down. So, so the extremophiles have two mechanisms. First, they have n number of DNAs, so that if one DNA is what to say, one DNA is broken down or degenerated because of any radiations, they have another set of DNAs inside them. Or the second mechanism is they have a quite good DNA repairing mechanisms. So I don't know any enzyme that actually shields them from the UV rays. The tardigrade you're talking about is has a ton state that or a tune state that it goes inside when the nutrients are depleted, and the insides of the tardigrade are frozen, as if frozen, or happens to go into cryo state. So I don't know any enzyme that actually blocks the radiation rather than which you are suggesting.
So to answer your question, there has been research on tardigrades, and they have a protein named DSUP or DSUP. I'm not sure if I'm pronouncing it correctly, and it basically forms a protective cloud against extreme survival threats such as radiation damage. So that is what protects them from the harmful UV rays.
I have also a question. So basically, like, they will eventually die, and I think what he meant was that, like, so basically enzymes are created mainly also from DNA. Well, basically also the function is going to change. So how does this even work? I mean, it can. I mean, they will even. I mean, eventually also enzyme is going to stop working at some point.
I'm sorry, but could you please repeat your question?
Sure. So basically speaking, enzymes are going to eventually stop working. What's your plan at that point? Or even before, actually, before when you have them. So basically, all these radiations are going to affect the DNA, which means also affect the production of enzymes, which also will affect the function of it. So I just don't see how it's actually going to work, more or less. But I mean, let's say it works and now eventually they die. What's your idea? How do you want to replace them?
So for our research, we are actually proposing that we use a combination of 2 to 3 microorganisms. We have also mentioned some of them in our report again. As far as this is concerned, there are some microorganisms which actually expand or grow under radiation. So as they are facing the UV rays, they are also growing along with it. So as some of them may not be as effective, but the fact that they are growing makes them stronger or retain their strength.